Assalamualaikum warahmatullahi wabarakatuh. Today I will be presenting my final year project titled A Review on Seaweed, the Prospect of Antibacterial and Antioxidant Properties of Extracted Caulerpa lentillifera essential oil. My name is Muhamad Akmal bin Abdullah and my supervisor is Madam Shafinas binti Abdullah. This is the content of my presentation today. Without any further ado, let's get deeper on this topic. Let's take a look at the background of the study. Seaweed contains excellent sources of nutrient and it has different vast and rich sources of protein, mineral, vitamin and bioactive compound. Caulerpa lentillifera is a species of seaweed which is rich in vitamin A and C, polyunsaturated fatty acid, mineral, essential amino acid and it has a low level of lipid. What are the problems that lead to this research? The first one is the current synthetic antibacterial and antioxidant available have been reported to be toxic and carcinogenic and it might affect the human health. The second one is knowledge about the benefit of Caulerpa lentillifera species as antibacterial and antioxidant properties has not been fully explored by researchers. And the third one is Caulerpa lentillifera contain high amount of water and low level of lipid, thus the best method of extraction should be explored to enhance the amount of essential oil. Next, let's take a look at the significance of this study. What are the significance of this study? The study helped to solve the health concern caused by synthetic antibacterial and antioxidant medicine. It is also a great opportunity to study and explore the antioxidant and antibacterial properties present in Caulerpa lentillifera species. What are the purpose of this research? The purpose of this research are to determine the most appropriate extraction method to extract the Caulerpa lentillifera essential oil, to identify the potential bioactive compound of Caulerpa lentillifera essential oil that can act as antibacterial and antioxidant agent, to observe the qualitative and quantitative determination technique of antibacterial and antioxidant properties of Caulerpa lentillifera extract. Now let's take a look at the research question for this study. The research question for this study are: what is the suitable sample preparation and extraction method of Caulerpa lentillifera sample? What are the potential bioactive compound of Caulerpa lentillifera essential oil that can act as antibacterial and antioxidant agent? How to observe the antibacterial and antioxidant properties of Caulerpa lentillifera essential oil? And what are the factors influencing them? Now let's take a look at the literature review of this study. According to JLR 2020, Caulerpa lentillifera is a type of green seaweed which belongs to the Caulerpa phylum, Overfica class, Porosopodil order, Caulerpaceae family, and Caulerpa species. It is typically freshly eaten raw or in a salt preserved state and sometimes used as ingredient for food and for feedstock by the local. It can be identified by their main features which are bright green color, a thallus with long horizontal and cylindrical stolon, straight red black front, and filiform rhizodal branches. Caulerpa lentillifera has great potential as antibacterial antioxidant due to its unique metabolite, but this species is highly underexploited compared to other flora. Now let's take a look at the proximate composition analysis. From the proximate composition analysis, fresh sample of Caulerpa lentillifera had a higher moisture content compared to dry sample, and dry sample had a better and higher as crude protein, crude fiber, and carbohydrate content compared to fresh sample. The crude lipid was found to have small difference between fresh and dry sample. This composition of Caulerpa lentillifera indicates that Caulerpa lentillifera have a high and beneficial nutrition content for human health. Now let's take a look at sample preparation of Caulerpa lentillifera. Pre-treatment of seaweed can be divided into two categories. The first one is pre-treatment of seaweed taken from seaweed habitat after collection, and the second one is preparation of pre-treated sample. Caulerpa lentillifera sample will be cleansed and rinsed with running water, either tap water or deionized water, in order to remove amplified sand and contaminants. And then it will be rinsed again for another 3 times using deionized water. In most cases of sample preparation, Caulerpa lentillifera sample will be either prepared by using sun drying, wind drying, thermal drying, and freeze drying method. Freeze drying method demonstrates better extraction of Caulerpa lentillifera extract compared to other sample preparation method. Is due to the physical damage of Caulerpa lentillifera cell by ice crystal formed during freeze drying might favor the extraction efficiency, and due to the fact that high temperature might induce oxidative condensation of some component and cause decomposition of some phenolic compounds. Next is the extraction of Caulerpa lentillifera essential oil. The most suitable extraction technique is needed to preserve the quality of final compound and to make the process achievable on industrial scale. The most preferable extraction method used during extraction of bioactive compound of Caulerpa lentillifera is salt extraction method due to its low cost and simplicity of operation. The extraction in salt apparatus is now the most commonly used method because of its ease of use, safety, scalability, simplicity, as well as it is cheaper and efficient option to extract the bioactive compound from the seaweed. Previous study proved the potential of antioxidant properties among seaweed families able to protect them due to the presence of hydroxyl group. One of the frequently used methods to measure the antioxidant activity is by using the FYP assay. FYP assay measures total antioxidant activity by calculating total reducing power from the ability to reduce triiodothiazine Fe3 complex to triiodothiazine Fe2 complex. When the Fe3 plus ion was reduced to Fe2 plus ion, an intense blue color will produce, which indicate that the reduction had took place and any changes in wavelength at 595 nanometer are monitored. Other methods to determine the antioxidant activity are by observing the total phenolic content and total flavonoid content in the sample. TPC was determined by using Folin-Keltu method, while TFC was determined by using aluminium chloride calorimetric method. TPC and TFC absorbance were measured and determined at 760 nanometer and 510 nanometer respectively. The method that can be frequently found in many literature for measuring the antioxidant activity is DPPH radical scavenging method. The PPH method is based on the determination of the reduction potential of antioxidant toward DPPH. The reducing potential can be measured by measuring the decline of its absorbance. The absorbance of the mixture will be measured and determined at 517 nanometer by using spectrophotometer. Let's go deeper into the antioxidant activity of Caulerpa lentillifera extract. The most frequently used antioxidant technique are the PPH radical scavenging assay and total phenolic content due to its simplicity and effectiveness in deciding the antioxidant properties of Caulerpa lentillifera extract. Result of TPC and DPPH value should present a positive absorbance value to show the ability as antioxidant. DPPH value was recorded in EC50 and IC50. The low in value indicates a high antioxidant activity. In reference to DPPH assay result in previous study, low EC50 and IC50 result was obtained, which indicate high efficiency of antioxidant activity. IC50 was more preferable compared to EC50. The compound responsible for antioxidant activity for Caulerpa lentillifera extract still remain undetermined. Based on previous study, the suggested potential compound to act as antioxidant agent are vitamin, C and E fatty acid, MUFA and PUFA, polysaccharide, sulfated polysaccharide, and phenolic compounds. Next, we have the antibacterial activity of seaweed. The antibacterial properties of seaweed are able to protect them by attacking the cell wall of the bacteria, thus injuring, killing, and preventing from further damage. This diffusion susceptibility assay. The bacteria often used for this technique are gram-positive bacteria and gram-negative bacteria. Firstly, agar plates are inoculated with a standardized inoculum of the tested microorganism. Then, on the agar surface, filter paper discs containing the tested compound at the desired concentration are placed. The diameters of inhibition growth zone are measured then. According to Bauer et al., the degree of inhibition will be expressed as follows: strong activity, inhibition zone more than or equal to 20 mm, moderate activity, inhibition zone less than or equal to 20 mm, weak activity, inhibition zone less than or equal to 12 mm, and there is no inhibition. The most frequently used antibacterial technique is this diffusion susceptibility assay in deciding the antibacterial properties of Caulerpa lentillifera extract. The Caulerpa lentillifera extract that had been tested appeared to be more effective against some gram-positive bacteria, for example Streptococcus mutans, than some positive bacteria such as Staphylococcus aureus and most gram-negative bacteria. The compound responsible for antibacterial activity for Caulerpa lentillifera extract still remain undetermined. Based on previous study, it is suggested that the responsible compound to act as antibacterial agent are alkaloid and fatty acid compound. Result of this diffusion susceptibility assay should be presented a positive inhibition growth zone to show the ability as antibacterial. What are the factors affecting the antibacterial and antioxidant activities of Caulerpa lentillifera? According to Osman et al. 2018, the external factors affecting the antioxidant and antibacterial activities of Caulerpa lentillifera include geographical location, salinity and depth, exposure of sunlight, and nutrients. Moreover, the technical factors are the type of solvent used for extraction and the sample extraction technique used. Next, we will move on to the conclusion and recommendation part. All in all, extraction of Caulerpa lentillifera extract was still carried out by using conventional extraction method, which is salt-slide extraction due to its simplicity, low cost and ability to scale up. For the antioxidant assay of Caulerpa lentillifera extract, most of the technique used for antioxidant assay are total phenolic content and the ppH radical scavenging assay, and it has been proven that Caulerpa lentillifera demonstrate the antioxidant properties. For the antibacterial assay of Caulerpa lentillifera extract, the most frequent used technique is the Day's diffusion susceptibility method, and extract of Caulerpa lentillifera have shown positive result on the antibacterial activity. The main compound responsible for the antioxidant and antibacterial properties remain undetermined and still need further study to get a fully grasp on the compound. There are many factors affecting the antibacterial and antioxidant activity of Caulerpa lentillifera, including the environmental factors and technical factors. In conclusion, Caulerpa lentillifera is a type of green seaweed species that have a wide potential of antibacterial and antioxidant agent. For the recommendation, there are not too many up-to-date research on Caulerpa lentillifera species in terms of its biological activity potential, as it remained underexploited throughout the years. It is suggested that many researches and studies should be done on the potential of Caulerpa lentillifera species, as it have a broad biological potential that can be explored in the near future. This is the cited reference used for this study. That's all from me. Thank you. Assalamualaikum.